السلام عليكم، مرحبا، أنا الدكتور مخلد جنابي. Today we will discuss the physiology of vision. This is the first lecture in the physiology part of the neuroscience module. First of all, let us see different parts of the eyeball. This part is the cornea. Behind it is the aqueous humor, and then we have the lens, and behind the lens we have the vitreous humor. All these structures are transparent media. In other words, they can transmit the light through them. And this transmitted light must be fallen on the retina to stimulate the photoreceptors there. Our visual system is dealing with light reflected from the object. So it is important to know some facts about light energy. As you can see, the light can be described as electromagnetic radiation, and our system can see certain wavelength of this radiation. The visible spectrum lies between 400 and 700 nanometer. Below 400 we can see it, above 700 we can see it. We can see only these wavelengths of 400 to 700 nanometer in length. Below 400 is called ultraviolet. We can see it, as I said. Above 700 is called infrared, and this is also invisible light. The other important things about the light, it moves in the space and in the air at very high speed, as you can see. The speed of the air, the speed of the light in the air, sorry, is about 300,000 kilometers per second. Very important in our system, this property of the light. The light, when it moves from one media to another media, the speed of the light will be reduced. And most important than this, if the light rays striking angulated object or surface, it will be refracted, as you can see. And this is what's happened when the light strikes the cornea and the other transparent media. The light is refracted, and this refraction of the light will ensure that the light will be fallen on the retina. The degree of light refraction by different structures is expressed by refractive index. And as you can see, the refractive index of a media equals to the light speed in the air over the light speed in that media. If we translate this to our transparent parts of the eye, we can see that cornea, aqueous, and vitreous and lens almost they have equal refractive index. Nevertheless, most of the light refraction in the eye occurs at the cornea, at the ear-cornea interface. And this explains why in the cornea the light refraction is the most, because the light is moving from the air where the media is of low density to cornea of high density. The light refraction by lens can be done by many types of lenses. These are the three types of lenses used to correct the error of refraction in the eyes. This one, as you can see, is called convex. And the light, as you can see, is accumulated in this point. From here to here is called focal length. This is a concave. The concave lens actually diverges the light rays rather than to converge it. And this type of lens is called cylindrical. The cylindrical can be either concave or convex, and you can see this one is convex cylindrical lens, and the light rays are, are focused in a line rather than on or in a point. The power of refraction of the lens is expressed in a diopter. It's a term used, and this diopter equals to 1 over the focal length of the lens in meter. So let us take example. If this focal length is 1 meter, the power of this lens is 1 diopter. If it is 10 centimeter, the diopter or dioptric strength of this lens is 10 diopter, and so on. This slide is about the eye as optic system. We have seen that we have 4 transparent media, and all these transparent media can transmit the light, and at the same time they can refract the light, and this refracted light must be fallen on the retina in order to see the object correctly. If we take all these structures and consider them as one single lens to measure the refractive power of the eye, we apply the law of diopter. If you remember, the diopter equals 1 over the focal length in meter. The distance from here to here is almost 17 millimeter. So if we divide 1 over 0.017, it gives you 60 diopters. This is the total refractive index of all transparent media of the eye. The refractive index of the transparent media is fixed except that of the lens, because the curvature of the lens can be changed. It can be increased or decreased. And by this increase or decrease in the curvature, the refractive power can be changed. Two important factors that determine the ability of the eye to focus the seen image on the retina are the length of the eyeball and the strength or the curvature of the lens, the degree of convexity. Changes in these factors lead to error of refraction, and we have 2 types of error of refraction in this regard. One is called myopia, and as you can see, in myopia we can see near object and we have difficulty in seeing the far object, while in hyperopia or hypermetropia or far-sightedness, the person with this error can see the far objects, and that person has difficulty in seeing near objects. In refractive errors, the light rays are not falling on the retina, as you can see here in this slide. In hypermetropia or hyperopia, the light rays, as you can see, falling behind the retina, while in myopia, the lights will fall in front of the retina. And both of these conditions will lead to difficulty in seeing certain objects. To compare these errors, just see this normal eye. It's called emmetropic eye. You can see the light rays are fallen on the retina. This slide is about the causes and the correction of the refractive errors. Myopia, the causes of myopia, uh, either the lens is too strong, the curvatures is more than normal, or the eyeball is long. And because of these factors, the light rays will be fallen in the front of the retina. To correct this error, we must use concave lens in order to bring these light rays to be fallen on the retina. The hyperopia or far-sightedness, we have seen that the image is fallen behind the retina, and the causes for this, either the eyeball is very short or shorter than normal, or the lens curvatures is less than normal. In order to correct this error, we must use convex lens. And convex lens, if this convex lens is put in front of the eye, it will bring this light rays to be fallen on the retina. We have another two types of refractive errors. One of them is called astigmatism. Its cause is unevenness in the curvature of the cornea. And this unevenness leads to falling of different planes of the image on different distance, and the image will become blurred. We can't see it correctly. The correction of this error can be done by using cylindrical lens. The astigmatism is very common. The other error is called keratoconus, means bulging of the cornea. The shape of the cornea is abnormal. It is bulging instead of this shape. As you can see, it is bulging forward. And the correction of this type of error is very difficult. It can be done by using contact lens. This slide shows the focal points of different planes of the image fall on different distances, and this will lead to blurring of the vision. So you can see this is the vertical light rays or the plane. The light is focused here, while the horizontal plane, the light coming from the horizontal plane, focused here, and this will lead to blurring of vision. By accommodation, we mean a rapid change in the ability of the eye to focus object at different distances, and this is done through changes in the curvature of the lens. As you can see here, the curvature of the lens is increased. This is done when we want to see near object, and in this eye the curvature is less. This is used to see far objects, and the ability to change the lens convexity, this will lead to change in the refractive power. the dioptric strength of the eye. And in young people, the curvature of the lens can be increased and the dioptric strength of the eye can be increased 14 diopter. So the refractive power of the lens inside the eye is about 20 plus 20 diopter. By increasing the curvature of the lens maximally, this power can be increased to plus 34 diopter. This is in young people, not in old people. Mechanism of accommodation. We remembered by accommodation, we mean that the eye has ability to see objects at different distances from the eye. And this process of accommodation is done through a ciliary muscle. And in order to understand this mechanism, we have to understand the relationship of ciliary muscle to the lens. From this picture, you can see ciliary muscle is attached to what is called zonule or suspensory ligaments. And these suspensory ligaments are attached to the capsule of the lens. If these suspensory ligaments are tight, they will pull on the capsule of the lens and making the lens less curved. On the other hand, if the ciliary muscle contracts, the zonule tension will be decreased. And a decrease in the tension in the zonule will release the tension on the capsule of the lens, and the lens become more convex shape or assume convex shapes. And this shape is useful to see near objects. And when the lens is less curved, this is useful to see far objects. And by this mechanism, we can see objects at different distances from our eyes clearly. This slide shows the accommodation mechanism in more details. So let us see this part of the picture. When the ciliary muscle relaxed, as you can see, the suspensory ligaments are pulling on the lens, on the capsule of the lens, and the lens become more flat. If you compare it to this side of the picture, when the ciliary muscle contracts, the suspensory ligaments become not tight, and because of less tightness in the suspensory ligaments, the lens becomes more round, and this is needed to see near objects, and less curved lens is needed to see far objects. This slide shows the difference between accommodated eye or lens and unaccommodated eye. So let us start by accommodated eye. When you want to see something near to your eye, you have to increase the curvature of the lens, and this is done through contraction of ciliary muscle. And the contraction of the ciliary muscle will lead to relaxation of the suspensory ligaments or zonule fibers. So releasing the tension on the lens or lens capsule, the lens will assume more curved shape. And this is good to see near objects. On the other hand, an accommodated lens is done through uh, relaxation of ciliary muscle. Relaxation of the ciliary muscle lead to increase in the tension in the suspensory ligament. Increase in the tension of suspensory ligament will pull on the lens, making the lens less curved or more flat. And this is good, or this is needed to see far objects. Accommodation is an active process, and this process is accomplished through contraction of ciliary muscle. Ciliary muscle contraction can be done through stimulation of parasympathetic nerve fibers to the ciliary muscle. And accommodation or contraction of the ciliary muscle is done by reflex action. It's called accommodation reflex. And because it is a reflex, we have to think about the circuit of this reflex. I mean, we need receptors, we need sensory fibers, we need integrating center, we need motor fibers, and the effector. So let us see this circuit. Sensory stimulus is the blurred image of the near objects on the retina. I mean, the receptors in the retina will see that near objects is blurred. And this blurred image will be detected or will be carried by sensory fibers through the optic nerve to the midbrain, and from midbrain through Edinger-Westphal nucleus through the parasympathetic fibers to ciliary muscle, causing contraction of the ciliary muscle. So as you can see, it is a reflex. It starts from the eye. The sensory impulses carried by sensory fibers in the optic nerve going to the midbrain, and then from the midbrain through the parasympathetic fibers to the ciliary muscle. Other facts about accommodation. Every time the eye is accommodated, there will be a convergence of the eyeball, and there will be a constriction of the pupil. It's called miosis. The constriction of the pupil is called miosis. The ability of the eye to accommodate is called presbyopia. And this condition happened or occurs when the person become old. This condition, presbyopia, is due to mainly due to two reasons. First, a decrease in the lens elasticity. The second, hardening of the lens. When the lens is not accommodated, the person cannot see or cannot focus on near objects, and the details of the near objects cannot be seen correctly. The decrease in the elasticity and hardening mostly due to deposition of insoluble protein in the lens, and this insoluble protein lead to hardening of the lens, and the elasticity of the lens will be decreased. When presbyopia occurs, the person, she or he, has difficulty in reading fine prints, headache, and visual fatigue. To correct this presbyopia, we can use convex lens for near vision. They call it reading glasses. The pupil. The pupil is an opening formed by the iris. The iris is the colored part of the eyeball, this one. and the iris consists of connective tissue and muscles. The opening of the pupil or the diameter of the pupil can be changed. It can be increased in this case or decreased when the pupil, this is called constricted pupil, this is called dilated pupil. Definitely, if the pupil is dilated, the amount of the light going through the eye, reaching the retina, will be more than when it is constricted. And they found that the amount of the light enters through the pupil is proportional to the square of the pupil diameter. The diameter of the pupil ranges between 1.5 millimeter, the diameter when it is maximally constricted, to almost 8 millimeter when it is fully dilated. Of course, there is personal variation between different peoples. So let us see how much the degree of the light enters the light when the pupil is maximally dilated compared when it is maximally constricted. So 1.5 square root of it and 8 square root of it. This is maximally dilated, this is maximally constricted. And this give you 2.25 over 64. It means that the amount of the light going through the pupil when it is maximally dilated will be more than the light entering the pupil when it is maximally constricted by 30 times. There is a 30 times increase in the light, amount of the light enter the pupil or enter the eye, reaching the retina when it is maximally dilated compared to when it is maximally constricted. The diameter of the pupil is controlled by two sets of the muscle. One is like a sphincter circulating the pupil, and this is called sphincter pupillae. Its contraction makes the hole smaller. On the other hand, there is another set of muscle. This muscle, or when it contracts, the diameter of the pupil is increased. This is called dilator pupillae muscle. The control of pupillary diameter is active process, is done through a contraction of muscle as we have seen in the previous slide. We have two sets of the muscles, one dilator, one is constrictor. The constrictor muscle is supplied by parasympathetic fiber. So activation or stimulation of parasympathetic fiber to the constrictor pupillae lead to constriction of the pupil, and this is called meiosis. On the other hand, stimulation of sympathetic fiber to the dilatory pupillae muscles lead to dilatation of the pupil, increase in the diameter of the pupil. This is called mydriasis. The condition is called mydriasis. The pupillary diameter control is a reflex action, and the stimulus for this reflex is the light intensity falling on the retina or reaching the retina. Formation and circulation of aqueous humor. The aqueous humor is formed by a structure, very vascular structure, is called ciliary epithelium. Is found in this place, as you can see. attached to the ciliary muscle or next to the ciliary muscle. That's why it is called ciliary process. It is very vascular structure producing aqueous humor. Once the aqueous is produced, it can pass from this area through or between the iris and the lens, reaching to this area, and from this area it will be drained through a canal here. I will describe it just in a few minutes. The place where the aqueous humor is formed, or this space, is called posterior chamber, and this space is called anterior chamber of the eyeball. So aqueous is formed in the posterior chamber, and it is drained from anterior chamber. So let us see the circulation or drainage of the aqueous humor. This part is enlarged to this part, this one. As you can see, this is the cornea, this one and this one. And we have a canal, it's called canal of Schlemm. And the aqueous humor is drained into canal of Schlemm. And you can see there is a trabeculae between the anterior chamber or the aqueous in the anterior chamber and the canal of Schlemm. And the function of these trabeculae is to interrupt any debris produced or found in the anterior chamber. And from canal of Schlemm, the aqueous is drained into aqueous vein, and from there to general circulation into the venous blood. The amount of aqueous humor in the eye in this area is about 125 microliter. This slide explains the formation and circulation of aqueous humor in details in a clear way. And as you can see, the aqueous humor is formed in this place from this structure. It's called ciliary process. Once the aqueous is formed, it moves into anterior chamber through the opening or the space between the lens and the iris, going to this angle, and it is drained through the Schlemm canal. This is the major drainage way, and there is minor one through what is called uveoscleral pathway. The major one is through the Schlemm canal. What are the functions of aqueous humor? The aqueous humor is found between the cornea and the lens, and it maintains the intraocular pressure. Or put it in this way, the intraocular pressure is due to presence of aqueous humor. The other function is maintaining the shape of the eyeball. The other functions, the aqueous acts as refractory media. And the fourth one, which is very important, it supplies nourishment to the lens and the cornea. Minding you, the lens and the cornea are avascular structure. So they get their nourishment, oxygen, and nutrient through the aqueous. The last function, it drains the metabolic end products. The metabolic end products produced through metabolism of the lens, the cornea, and the structures between them will be drained with the aqueous humor to the venous side of the circulation. Intraocular pressure. What is meant by intraocular pressure? It means that the pressure of the fluid in this place, the aqueous humor, and the pressure can be measured in practice, and the mean intraocular pressure in human beings is about 15 millimeter of mercury. If the intraocular pressure is increased, this increase in the pressure will be transmitted to vitreous humor and then will be transmitted to retina, or it will affect the retina. It will cause damage to the neural layer of the retina, destroying the neurons in the retina. Once these neurons are destroyed, uh, it may lead to blindness. This condition of abnormal elevation of intraocular pressure is called glaucoma. In this sentence about aqueous humor, where it is produced or it is produced in the posterior chamber and produced by ciliary process, and it is produced at a rate of 2 to 3 microliter per minute. This picture shows different layers of the retina, and the retina is the neural layer of the eye. Don't worry about the name of these layers of the retina, 10 layers. What I want you to know, the types of neurons, types of the cells in the retina. So if we start from here, this one, these cells or neurons called ganglion cells, and the axons of these ganglion cells forming the optic nerve. And this type of cells, the blue one, called bipolar cell. And these cells are the photoreceptor cells. So the light coming to the retina will go through all these layers until it reaches to the photoreceptors. The photoreceptor or the photoreceptors will absorb the light. The extra amount of the light which is not absorbed by the photoreceptors will be absorbed by this layer, the pigmented layer. This pigmented layer is very important because by the way, it contains a lot of melanin pigment. So if this pigmented layer is not found, the light which is not absorbed by the photoreceptor will be reflected back toward the eyeball. And this reflected light will cause a lot of trouble and cause or decrease the ability to see perfectly. One example for absence of this pigmented layer is found in unfortunate people, the albinos people. The albinos people, the visual ability, visual acuity of albinos people is very, very low, simply because of absence of pigmented layer or pigment in this layer, and the light will not be absorbed. The other function of this pigmented epithelium, pigmented layer of the retina, is the storage of vitamin A. And the storage of vitamin A is important part of the photopigment found in the photoreceptors. So every time we need vitamin A to synthesize the photopigment, some of this vitamin A is coming from pigmented layer of the retina. Let us talk about important landmarks in the retina. We have two important landmarks. Let us start by describing the first one, this one. As you can see, it is paler than the surrounding area. It is called optic disc. The optic disc is the area where the optic nerve uh, is coming to the retina, and it is also the area where the blood vessels coming to the retina and leaving the retina. This area sometimes is called blind spot simply because it uh, doesn't contain any photoreceptors. The optic disc is found on the nasal aspect of the retina. And temporal to the optic disc, we have another important landmark. It's called fovea. This fovea has the highest visual acuity. If you want to see image, the fine details of the image, this fovea will help in seeing these details. That's why it is called, or it has the highest visual acuity. The fovea contains a lot of the photoreceptors, mainly the cones, few rods, and in the center of the fovea, we have another very small area. It's called fovea centralis. And this fovea centralis contains only cones. This slide is about the fovea and why it is used to see the details of the image. In other words, fovea has the highest visual acuity compared to the other area of the retina. And the reason for this high acuity, as you can see listed here, first reason is the high cones density. It means a lot of cones here in this area. This is the photoreceptor layer. The density of the photopigments, especially cones, are very high here. The other reason, as you can see, the layers of the retina are pushed aside and the light pathway becomes short, so it reaches the photoreceptors easily. The other reason for high acuity of vision of the fovea is the absence of the blood vessels covering the fovea. And the last one is the special connection of the foveal cones, the photoreceptors. The number of the photoreceptors connected to one ganglion cell, here are the ganglion, are small number, and this will lead to an increase in the acuity of vision. This slide is about retinal blood supply. Retina receives blood supply through two sources. The first source is through the central retinal artery. The blood coming through the central retinal artery supplies the inner two-thirds of the retina. As you can see, this ganglion cells, and these are the bipolar cells. The outer third of the retina, the photopigment layer, is supplied through choroidal or choroid capillaries, as you can see. So the outer layer of the retina is supplied through the choroid. For this reason, if there is a detachment of the retina from the choroid, this is called retinal detachment. In this case, the blood supply or nourishment or blood supply to the photoreceptors will be stopped. And this eventually lead to death of the photoreceptor. For this reason, the retinal detachment must be replaced very quickly. Otherwise, we will lose the photoreceptors. If the photoreceptors are lost, it means blindness. This diagram shows the basic neural connections of the retina. And you can see the photoreceptor is attached to the bipolar, and the bipolar is attached to the ganglion cells, and the axons of the ganglion cell forming the optic nerve going to the brain. So this is the basic neural connection. If the bipolar cell is connected to rod only, then the connection must contains or must involve amacrine cells. Otherwise, this is the basic neural connection. This diagram shows again the connections of neurons in the retina. Uh, the basic, as we said, photoreceptor, bipolar, and ganglion. But you can see we have a horizontal cell connecting the cones to the bipolar cell, cones or rods, or both of them, to the bipolar cell. And we have amacrine cells in this layer of the retina connecting the bipolar cell to the ganglion cell. This diagram shows in details the interconnections between different types of neurons of the retina. We have seen that the basic structure or basic circuit of neurons consists of photoreceptor connected to the bipolar cell, and this bipolar cell connected to the ganglion cell, and the axons of the ganglion cells sending action potential to the brain for interpretation of the visual image. You can see the horizontal cells is found between the photoreceptors and bipolar cell, and the amacrine cells is found between the bipolar cell and the ganglion cell.